The next major type of membrane transport is active membrane transport. And this means that it goes up, it's a concentration gradient, and it involves energy, which is ATP. The two major types of active transport are active transport, and these involve ions. And a really good example of this is the sodium potassium ATPase transporter. Other types of active transport could involve other ions, but this is a super common one that we're going to see on the next slide. Vesicular transport occurs when there are large amounts of molecules. And there's so many molecules that the cell has to use a vesicle, which is essentially almost looks like a blob which is surrounded by a membrane. And that membrane is going to form this vesicle, thus bringing in a large amount of molecules or taking those large amount of molecules off. So both are going to require ATP, and the solute is too large for channels. It's not lipid soluble, and it's not able to move down its concentration gradient. So it requires ATP. So that's a significant difference between between passive and active. And it also goes against its concentration gradient. So those are the two big differences that you should know. The next slide is showing the sodium-potassium pump. So we're going to zoom in here to look at this, and it's called primary active transport because it directly utilizes the ATP for movement of this channel. So the first thing that happens is that 3 cytoplasmic sodiums bind to the pump, and that's on the intracellular fluid side. So we see one here, another here, and finally this is the third. So 3 on the inside. So this is kind of like a turnstile. So 3 sodiums are going to bind. The next thing that's going to happen is that the sodium binding promotes hydrolysis of the ATP, and it releases energy during this reaction. So just like you learned about in chapter 2, the ATP is broken down to ADP plus an inorganic phosphate. And this inorganic phosphate represents energy that's released. And it's enough energy that this channel can physically turn so that now that sodium can start to move to the extracellular fluid side, which is what we see in number 3. So those sodiums are now released on the extracellular fluid side. The phosphorylation causes the pump to change shape, expelling the sodium to the outside. So again, think of that example like a turnstile. The next step is that now 2 extracellular potassiums are going to bind. So we have 1 potassium here and another potassium here. Step 5 is we're going to see the potassium triggering the release of a phosphate, and then the potassiums are released on the inside. And we also see another ATP broken down into ADP. So the potassium is released on the inside. So the summary of this is that basically 3 sodiums exit the cell and 2 potassiums enter the cell. And this requires ATP for this to happen. So this is the most common example of active transport. Now, when the energy is indirectly used, it's called secondary active transport. So the word secondary really means indirect. So energy is stored in that gradient from the sodium potassium pump, and that energy is then used for other things to happen. The sodium essentially drags other molecules with it, and it flows into the cell through carrier proteins. And it's usually symporters, but involves things like sugars, amino acids, or ions. So again, secondary active transport means that it's indirect and that it's the result of the sodium potassium pump. So the sodium potassium pump is direct, and this is indirect, which we're going to see on this slide. So in number 1, we see primary active transport, where ATP is directly used. It's directly broken down. Remember, we had 3 sodiums that went out, 2 potassiums that go in. So in summary, it's called the sodium potassium pump. Now, because this pump has occurred, there's others that can kind of go along for the ride. They can kind of go along with the energy that has been produced, that's been stored in the membrane as a result of the sodium potassium pump. One of those examples could be either glucose, or we could see amino acids. So in this case, the glucose is going to now cross. Since it's active transport, it's still going to go against its concentration gradient into the cell. So now, when we have more than just one ion that's going to cross, we need to use a blob, which is surrounded by a membrane. That blob is called a vesicle. So vesicular transport is when there are thousands of molecules that all move at once. So it's transport of large particles, macromolecules, or fluids across a membrane in vesicles. It still involves ATP, and there's 3 types based on whether it enters the cell or exits the cell and the type. So if it enters the cell, it's called endocytosis. If it exits the cell, it's called exocytosis. And if it goes across the cell, it's called transcytosis. And then there's 3 types of endocytosis. Phagocytosis, if it's a solid that's being transported into the cell. Pinocytosis, if it's a liquid, or a very specific type is called receptor-mediated endocytosis. And a lot of times this involves things like the LDL molecule, which is a type of cholesterol, oftentimes thought of as the bad cholesterol. So this slide shows us some of those examples of vesicular transport. So in the first example, we have endocytosis. You don't need to know the coded pit or anything like that. Just know that endocytosis is entering the cell. So we're going to call that endocytosis. Once the vesicles enters, as you can see here, we then have some that can exit the cell, and that's what we see here at this other end called exocytosis, or it can cross the cell as transcytosis, but it's always going to travel in a vesicle, and that's what's important. In this situation, we see that the contents of the vesicle are going to be destroyed, and that's using what's called a lysosome. And we'll talk about that later on in the chapter. So on this slide, it's showing the three types of endocytosis. So phagocytosis is a solid that it's bringing in. And that's what's important to remember about it. Pinocytosis, it's almost like the cell is gulping liquid, extracellular fluid, and it's nonspecific. So phagocytosis and pinocytosis are nonspecific. The one that's specific is receptor-mediated endocytosis because it's mediated by a receptor. This receptor is going to identify a very specific molecule. So let's say that these LDL or these green solutes are LDL molecules. This specific receptor, which remember is a protein, is an LDL receptor. So the receptor-mediated endocytosis is more specific.